Okay. Hello, everyone, and thank you all for joining us today. I'm very, very happy to be moderating this session of the RNA Collaborative Seminar Series as one of the organizers of the Cambridge RNA Club. And today we are hosting two very exciting seminars, of course, focusing on RNA. But before moving on to science, let me just remind you to please type in your questions in the Q&A chat, Q&A instead of the chat. So our first speaker, as you saw, is Anna Vidaković. Anna obtained her first degree at the University of Belgrade before moving to the CRUK Cambridge Institute for her PhD. During her PhD, she attended a talk in which a mention of transcription-coupled DNA repair inspired the direction of postdoctoral research at the Francis Crick Institute. There, she discovered how a single post-translational modification or RNA pol2 can regulate the cellular response to DNA damage inflicted by UV light. Her findings inspired further questions that she now aims to answer with her group at the MRC Laboratory of Molecular Biology in Cambridge. And today we are hearing more about her research in her talk, Ubiquitin-mediated control of RNA polymerase 2 and the onset of the transcription cycle. So please, Anna, over to you.
Thank you, Bianca. So I'm going to share my screen now. Okay, so yeah. Hi, everyone. Well, thanks to the organizers for this opportunity to present our recent findings, which show how ubiquitination of RNA polymerase 2 controls the beginning stages of the transcription cycle. So in our lab, we study RNA pol2, and this particular project was led by 4 different people, Roberta Cacioppo in my lab, Ivan Schlamovitz from our lab, Alexander Gillis from Scott Berry's lab in Australia, and Andrew Zeller from our neighboring Julian Sales lab. So as I said, in our lab, we study RNA polymerase 2 or pol2, which at the heart of the central dogma transcribes all protein-coding genes and thousands of non-coding RNAs. And we study pol2 primarily in human cells. So transcription begins when transcription factors recruit mediator and the pre-initiation complex, which then recruit RNA polymerase 2 from the free pool of molecule onto the DNA and position it at the transcription start site. And then a series of proteins not shown here will help to open the promoter DNA and allow pol2 to start initiating, so to start transcribing. But on most metazoan genes, or at least many metazoan genes, pol2 will not get very far because it will soon reach the pausing site where it will stop. Now, only after a coactivator, which involves a kinase, is present, it will phosphorylate pol2 and associated proteins not shown here, triggering conformational changes and release of pol2 into elongation. So this zone between transcription start site and the pausing site is often called the promoter proximal zone, and it is actually really small. It is only 60 base pairs long on average, but it is exactly in this very small space where most key decisions about a gene expression are made by controlling the rate of transcription initiation and the rate of pause release. And things are actually a bit more complex than that, because recently it has been found that 80% of pol2 molecules that initiate transcription never make it past the pausing site, but are instead prematurely terminated or aborted in the promoter proximal zone. So regulating any of these steps really can provide an opportunity to influence and determine gene expression. So today I'm going to tell you about a negative regulatory mechanism that we discovered works here to regulate pol2 in the promoter proximal zone. But before we go into that, I'll just like to give you a little bit of a context. So over the past several years, we have mostly been interested in elongating pol2, so the one that's already past the pausing site. And elongating pol2 can stall on obstacles inside genes. These could be DNA-bound proteins or secondary DNA structures or DNA damages, and these obstacles come from endogenous sources, so we always have them in our cells, but can be further amplified by exogenous sources like genotoxins, like UV radiation. So when Pol2 stalls in opticals, it literally cannot move any further, and it will block the path of all polymerases traveling behind it. So this is a serious problem, and we were interested to study how is that regulated. Now, what's interesting is that both paused Pol2 and stalled Pol2 have in common is that they're not moving, they're stopped, but as far as we can tell, they are regulated by completely different mechanisms. So what happens to stalled Pol2? Well, we discovered a few years back that stalled Pol2 is extensively regulated by ubiquitination. And you will all know that ubiquitination of proteins is a post-translational modification whereby a small protein ubiquitin is covalently attached to its substrate molecules. And this often happens in the forms of polyubiquitin chains that can have different linkages. And these different types of chains provide enormous regulatory potential. The most famous one you all probably know about are K48 link chains, which usually lead to protein degradation. And also, almost every biological process you can think of is regulated by ubiquitin at some level. So what about transcription then? So we know stalled Pol2 is regulated by ubiquitination. And we know that this ubiquitin is conjugated to a single lysine on Pol2, lysine 1268. Now, this event has many important consequences for the cell, but today, the most important to remember is that this is the only ubiquitination site on Pol2 with a well-documented and known function. So we started a new project when I started the lab, simply asking, is Pol2 regulated by ubiquitination in any other situation other than stalling at any other place on the gene? So to address that question, we took a very simple approach. We inhibited proteins that process ubiquitinated substrates. So P97 is a segregase that extracts subunits that are ubiquitinated from multi-subunit complexes, and then it feeds them to the proteasome that degrades them. So we blocked P97 and the proteasome in otherwise untreated cells and simply performed ubiquitin pull-down assay. So in this assay, we're using this DSK2 resin that binds ubiquitin on ubiquitinated proteins, pull them down, elute them, run them on a gel, and analyze them by Western blot. So when we do this for Pol2, we are blotting actually for RPB1, which is the catalytic subunit of Pol2. So I'll be using the name RPB1 and Pol2 interchangeably throughout my talk. And as you can immediately see, when we inhibit P97 in otherwise untreated cells, large amounts of polyubiquitinated RPB1 can be observed. And importantly, when we do the same experiment in lysine 1268 mutant Pol2 cells, so these are the cells where Pol2 cannot get ubiquitinated after UV, which causes stalling during elongation, but this mutant still gets ubiquitinated after P97 is inhibited, which means that these two forms of ubiquitinated Pol2 shown in purple and yellow are completely different forms. Therefore, we are looking at a new form of ubiquitinated Pol2 that is likely different than stalled Pol2. Now, I don't have time to go into details. We figured out that there is an additional protease involved in fragmenting this RPB1 protein before it's chopped up by the proteasome, but the point is that we believe this ubiquitination leads to Pol2 degradation. So we then wanted to understand where in genes does this happen? What is the ubiquitin ligase responsible? And what is the function of this process? So let's start with the first question. Now, Pol2 is dynamically phosphorylated during transcription. When it starts transcribing, it becomes phosphorylated on serine 5 residues in its repetitive C-terminal domain. And only after it is released from pausing, it accumulates serine 2 phosphorylation and loses progressively serine 5 phosphorylation. So we can use these states as a marker and blot for serine 5 and serine 2 phosphorylation in the ubiquitin pull-down assay. So what you can see is that after UV radiation, which causes elongation stalling, you see that this ubiquitinated Pol2 is primarily phosphorylated on serine 2 residues and a little bit on serine 5, which agrees with the fact that it happens here during elongation. But the form that is stabilized when P97 is inhibited only contains serine 5 phosphorylation and no serine 2 phosphorylation, which means that it must come from here, from the promoter proximal zone. So this was very exciting because regulating Pol2 levels here, as I've said, would have enormous consequences for the transcription cycle. So we then went on to find the ubiquitin ligase so that we can remove it and study the function of this process. And ubiquitin ligases come in different families, and this Karli ring family of ligases can be inhibited with this small molecule MLN. So when we apply MLN to cells, the ubiquitination of Pol2 that is stabilized by P97 inhibition is gone. So the ligase we're looking for must belong to this family. And then Karli ring ligases are modular enzymes that rely on 8 different Karli molecules that then bind different substrate recognition proteins to recognize hundreds, if not thousands, of substrates in a cell. So we wanted to know which one of the Karlins is responsible for this, and we performed siRNA knockdown of each individual Karlin and found that Karlin 3 based ligase is responsible for this Pol2 ubiquitination. Now, Karlin 3 binds over 90 different substrate recognition proteins, so this would have been a very large screening exercise, but we were lucky that around the same time we were doing this paper came out, which showed that RMC5 protein, which is a Karlin 3 specific substrate recognition protein, actually is mutated in this disease, adrenal hyperplasia. And when they did some molecular characterization of this protein, its top interactor is RPV1, so Pol2. And they found that RMC5 Karlin 3 indeed can ubiquitinate Pol2. But the function of this, or where on genes this would happen or why, remained unknown. So for us, this was, of course, the obvious candidate to then knock out, as we did, and perform the ubiquitin pull-down assay. And you can see that ubiquitination of Pol2 is now completely gone. So RMC5 is the ligase that targets promoter proximal Pol2 for ubiquitination. Importantly, I'm not showing the data, but as far as we can tell, ubiquitination of elongation stalled Pol2 does not have anything to do with RMC5. Okay, so now we found the ligase, which means we can abolish it and study the consequences for the transcription process. So that's exactly what we did, and we started by profiling Pol2 occupancy genome-wide using DXChIP-seq. And what you can see even from this one gene example is that Pol2 accumulates at transcription start sites. This is true across the genome. So basically, Pol2 accumulates at start sites of almost all genes in the genome, but not in gene bodies or transcription termination sites. So this effect is global, and almost all genes in the genome are affected. So this may be expected because we, in the previous slide, saw that RMC5 ubiquitinates Pol2s that are in this area of the transcription cycle. So when RMC5 is gone, Pol2 accumulates there. But what has surprised us is that when we looked beyond chromatin into the free pool of molecules as well, we did that using FRAP or using chromatin fractionation, we saw that Pol2 also accumulates in the free pool even more dramatically when RMC5 is gone. So this just reminds us that we need to look at transcription as a dynamic system. And I've told you that 80% of Pol2 molecules from the promoter proximal zone are aborted. So RMC5 regulates Pol2 at both of these initial stages of the transcription cycle. And further in the talk, I'll give you some understanding of how this happens. So we then wanted to understand, what does RMC5 actually target on Pol2? Why does it ubiquitinate Pol2? And one hypothesis we had is that it may target Pol2 molecules that are somehow defective or incompletely assembled. And the reason we thought this could happen is because the transition between initiation to pausing to elongation is actually really complex. Pol2 doesn't work alone. It assembles with many other factors that need to exchange on it. Pol2 tail needs to be modified, the RNA needs to be kept, and then Pol2 needs to be stably paused, released into elongation. So all of these exchanges are extremely complex. And we wondered, what if this sometimes goes wrong? And what if that triggers recruitment of RMC5 and Pol2 ubiquitination? So luckily, there are many inhibitors that can block these different transactions or capture Pol2 in some of these transient states. and we applied them and tested what will happen to POL2. So all of these inhibitors of different stages of promoter proximal transcription, all of them that we tested cause POL2 degradation, which is completely AMC5 dependent. And furthermore, when we use an expanded panel of inhibitors and look at POL2 ubiquitination directly, all of these inhibitors cause some level of POL2 ubiquitination, and all of that depends on AMC5 fully. So indeed, AMC5 can target perturbed early transcription complexes. So one of the treatments that really drew our attention is this triptolide, because it caused the most dramatic ubiquitination of POL2. So what is triptolide? Triptolide inhibits initiation. It inhibits this XPB translocase, which is facilitating promoter melting and the beginning of transcription. So in theory, when triptolide is applied, POL2 cannot move beyond the TSS. It cannot enter these other stages of transcription. So triptolide is also known to cause degradation of POL2, which you can see in this immunofluorescence. And what we find is that this degradation completely depends on AMC5. So in AMC5 knockout cells, there's no POL2 degradation. You can see that on the Western blot as well. But what was really shocking to us is that we looked at POL2 modifications. We saw that the marker of elongating POL2, saying 2 phosphorylation, also persists in AMC5 knockout cells. So how is it possible that, we inhibited initiation, but we're still getting POL2 to go into elongation, it seems? So to test this directly, we performed ChIP-seq again, and you can see that indeed, upon triptolide treatment in wild-type cells, POL2 is eradicated because most of it is degraded. But in the AMC5 knockout cells, a lot of POL2 still is present, not just on the transcription start site, but also in gene bodies. And actually, when we do nascent RNA sequencing, TT-seq in this case, we also see that this POL2 enters elongation and transcribes. So in the absence of AMC5, inhibited XPB can be bypassed and POL2 can transcribe and elongate into the gene. And as a consequence, the low doses of triptolide that would normally kill wild-type cells do not kill AMC5 knockout cells, so they're partially resistant to triptolide. What this means is that as long as POL2 is not first degraded by AMC5 after triptolide treatment, some of it can bypass inhibited XPB and proceed into pausing and elongation. And this is important because triptolide is often used as a tool to study POL2 dynamics in the promoter proximal zone. And the assumption of all of these models is that the inhibition of initiation is absolute, which clearly it's not. So we would suggest that these findings be taken into account for future studies modeling POL2 dynamics. Now, upon triptolide exposure, not all POL2 stays on chromatin. Actually, we find that around half of it or more is kicked out. It's evicted from chromatin. And you can see this in this time course of triptolide. You get less and less POL2 on chromatin and more accumulation in the soluble fraction in AMC5 knockout. And in wild-type cells, AMC5 will simply degrade this POL2. So we also see that this POL2 that is kicked out is phosphorylated. So that really means it came from the DNA, because as far as we know, POL2 should only be phosphorylated on chromatin while it's transcribing. And importantly, even in untreated conditions, there is accumulation of phosphorylated POL2 in the soluble fraction, so in the free pool of molecules where it shouldn't exist in AMC5 knockout cells. So this gives us some more idea of where AMC5 could work or what it could target. Basically, if these molecules are in constant rounds of initiation and premature termination, we believe that AMC5 could be working here, targeting molecules that were evicted from the DNA, prematurely terminated. And this is supported by just long exposure of this experiment, where we detect ubiquitinated POL2 traces only in the soluble fraction, but not on chromatin, which supports the idea that AMC5 works here. Now, regardless of where exactly it targets POL2 molecules, the fact remains that each successive step of the transcription cycle progressively buffers the effect of AMC5 loss. What this means is that in the free pool of molecules, which we can measure by FRAP as a mobile fraction or soluble fraction in chromatin fractionation, different assays, but the same result, there is around twofold increase in Pol2 molecules in the free pool. There is around 40% increase of Pol2 molecules at transcription start sites in AMC5 knockout relative to the wild type, but there is no increase of Pol2 in gene bodies or no increase in nascent RNA activity, nascent RNA production, so no increase in elongation, really. So we were wondering, what is it that prevents this excess Pol2 at transcription start sites in the RT5 knockout from being released into the gene body? So there is clearly something stopping this accumulated Pol2 to proceed into the gene. So we reasoned because AMC5 knockout cells on its own, they don't have any notable phenotype. We reasoned that if we remove that factor that is gatekeeping the release of these promoter proximal Pol2 into the gene, that might be lethal to the cells. So we could actually perform a synthetic lethality screen. And we systematically knocked down most of the factors that are well known to operate in the promoter proximal zone and regulate Pol2. And we found a strong synthetic growth retardation phenotype when integrator 8 was depleted in AMC5 knockout cells, as you can see on this orange line. So what is INTES8? What is integrator? So INTES8 is part of integrator phosphatase module, which has recently been shown to counteract the activity of coactivators that contain CDK9 kinase. So basically, this complex dephosphorylates Pol2 and associated factors, and the activator tries to phosphorylate it to promote post-release. So essentially, the phosphatase is preventing post-release. So we then went on to test how the combined loss or individual loss of AMC5 and integrator affects Pol2 activity on genes. So when we look globally across the genome, we don't see any synergistic effect of these two factors when we compile all the genes together, but we do detect integrator-dependent increase in nascent RNA production in the beginnings of genes, at least, and this was reported before. However, when we zoom into different gene classes, which we routinely do, we find that short genes have a very profound synergistic effect of AMC5 and integrator 8. When both factors are removed, as shown in the orange line, these genes get, on average, upregulated. So this is just because we routinely do this classification, but we wanted to unbiasedly test what kind of genes are affected by integrator 8, AMC5, and both in combination. So we performed differential gene expression analysis. We found genes that are regulated by these factors, and then we did various analyses to see what is so special about these genes. And I'm not going to go through all the data, but the most significant association we find was the baseline expression of these genes. Basically, when AMC5 or integrator 8 are removed, or both in combination, genes that have a very low starting level of expression, very low baseline expression, either get reactivated or further suppressed. And we find these genes also tend to have Tata boxes in their core promoters. So this is easier to grasp when we really look at individual gene examples, as you can see here. So removal of integrator does result in upregulation of many of these genes, as was reported before. But in AMC5 knockout, because now we have accumulation of Pol2 at the transcription start sites of these genes, when we also remove integrator, this excess Pol2 is unleashed into the gene, which results in gross upregulation of these genes. So this is our model, where we really think that AMC5 regulates and surveys the quality and quantity of Pol2 molecules, so transcription complexes, prior to post-release, so at the beginning stages of the transcription cycle. And then integrator 8 acts as the next checkpoint or a gatekeeper that prevents the excessive release of these complexes into elongation. So with this, I would like to end and thank you for your attention and thank all the members of my lab for doing this amazing work. So Roberta Cacioppo and Ivan Šlamovic led this work from my lab. Alexander Gillis led the work from Scott Berry's lab, and Andrew Zeller did the analysis. He's from Julian Sales' lab, who's next door, and also Alistair, Angela, Yubal, Pega, Ben, and Daniela also helped significantly on this project. And we will soon be recruiting a computational biologist. So if you're interested or know someone who is, please apply or spread the word. So thank you for your attention. I'll be happy to take your questions.
Thank you, Anna. Thank you for the great talk. While people start to type in their questions, maybe I can start with a few curiosities that I have. So I was wondering if there is any link. I'm not in the kind of field of RNA Pol2, but expert. But if I was wondering if there is any link between the type of promoters that Pol2 transcribes and the modification that it carries with it.
That carries with it. That's an interesting question. I think the SERIN 2, SERIN 5 code should be relatively universal. But actually, there are other modifications on Pol2, and they're very poorly studied. So it could be that this is affected. But then you can also take into account modifications we don't even know exist yet, like this one. We didn't know this happens to Pol2, this new ubiquitin type. And that, while it happens globally, it has more profound effect on genes that have tata boxes that are locally expressed. So.
Yeah. Thank you. And just another one. I guess you are working with human cells. Is that correct? Okay. So do you have any idea? I mean, I don't know, but probably you know better. And do you have any idea of how well the ubiquitination of RNA Pol2 is like conserved across species?
So the ubiquitination of stall Pol2 is conserved even in yeast. So that's a very conserved process. And as for AMC5, I mean, we know that mice that have this mutation in AMC5 develop the same disease in humans. And this nice paper by Lau et al. Showed that they accumulate excessive Pol2 in almost every organ they've looked at. So at least in mammals, this is a conserved process that can lead to disease when it's mutated.
Okay. Thank you. I'll read some of the questions now. So we have one from Nicholas. In your UB pull down, some of the RPB1 doesn't appear to shifted. Are these just lowly UB or are there some other subunits UB?
Yes, so that's always RPB1, so that's the subunit we're looking at. And the band that is not shifted, people have called that monoubiquitinated Pol2. But actually, because, you know, the washing of the beads can never be 100% perfect and you have a lot of input, it could be just the carryover of unmodified Pol2, which is actually quite useful to have as a reference point. But we will be doing assays to actually finally put an end to that question. Is that monoubiquitinated Pol2 or is it a carryover of input, really, not just the unmodified Pol2?
Thank you. We have a few ones. When it was shown the ubiquitination status of RBP1 upon triptolide, it showed increased levels, but as I understood, RMC5 is responsible for ubiquitinating RBP1 in promoter proximal. So is there another ligase involved or perhaps another ubiquitin system protein?
So when we say promoter proximal, we mean everything from the TSS to the posing site. So the triptolide freezes Pol2 in theory, at least at the TSS, but RMC5 is fully responsible for degrading this Pol2. So when we knock out RMC5, there's no degradation whatsoever. So we think that's the direct ligase. But what I was talking about is that we're not yet sure where exactly this ubiquitination happens. Is it on the DNA when Pol2 is locked in these defective states, or does Pol2 actually fall off first and then is degraded and cleared in the soluble fraction? So we don't think there is another ligase involved, at least in these effects that we were talking about today, but there could be other ligases acting on genes at different parts of the transcription cycle. We're working on that too.
Okay, thank you. I'll read the last one and maybe you can keep on answering in the Q&A. So do you know which residue is ubiquitinated by RMC5?
We have tried to find that and we didn't manage. So basically we know that, well, when we mutate this lysine 1268, which is the ubiquitination site after DNA damage, after UV, so when Pol2 stalls, ubiquitination by MC5 still happens. So this does not seem to be the same site. But when we tried to do mass spec, we really couldn't identify yet. And that could be just to technical limitations of the methods. We are also trying to improve them to make them more sensitive to see if we can detect the sites using some different methods. Yeah.
Okay. Cool. Thank you. So yeah, I will move on with the second speaker.
Okay. Thank you.
And you can, yeah, thank you for your talk. Amazing. So our second speaker today is Kostas Tzelepis. He graduated from the University of West Attica in Greece with the BS in Biomedical Sciences. He then got his master from the Medical School of the National and Capodician University of Athens in cancer biology. After this, he moved to Cambridge, where he got his PhD in molecular genetics from the Wellcome-MRC Institute, working on developing one of the first genome-wide CRISPR screening platforms for the identification of cancer vulnerabilities and novel therapeutic targets. He has been a Wellcome Trust Sir Henry Dale Fellow at both Harvard University and University of Cambridge, before becoming a group leader at Milner Therapeutics Institute in Cambridge. And now he recently moved to Cambridge Stem Cells Institute, where his research focuses on RNA modifications. And we will hear more about this in his talk today. Whose title is Therapeutic Targeting of RNA and its Modifications. So over to you, Kostas. Thank you.
Thank you very much. Let me share my screen so that we can kickstart. Right. Can you see it? Yes?
Yes.
All right. Let me... Okay, so thank you very much for the lovely invitation. So our lab, as you mentioned, is based at the Cambridge Stem Cell Institute at the University of Cambridge. We are focusing on the role of RNA modifications in the context of stem cell homeostasis and disease. These are our latest studies, collaborative studies, including a phase 1 clinical trial, which is ongoing. And the general vision of our lab is to uncover novel fundamental RNA biology while we are trying to push our discoveries towards therapeutic translation. And I will show you a couple of examples today. So the general theme of the talk is the RNA modifications and the epitranscriptome, which, as many of you know, it's a pervasive new mechanism of metazoan biology and gene regulation. There are over 170 RNA modifications reported, most of them underexplored. It's fair to say this is a largely underexplored field of research, but lately, recent studies, including some of our end, have shown that epitranscriptomic pathways are implicated in stem cell homeostasis and disease, and with many focusing on cancer as well. So the first part of my talk is going to focus on M6A. This is one of the most abundant RNA modifications in eukaryotes. We know that this modification has writers, readers, and erasers. Many of those are reported, including METL3, METL16, and METL5 in terms of the writers. We know many of the readers. It used to be the YTH domain containing proteins, but there are many more reported during the last few years. And we also have the erasers, including LBH5 and FTO, although they have other functions beyond M6A. So one of our recent studies show that METL3, the M6A writer, is recruited via the transcription factor CPPZ on the promoter of a subset of genes, is installing the M6A modification within the coding sequences, and then that the position of the modification is actually boosting mRNA translation of oncogenic transcripts, and the removal of that modification via METL3 is actually causing mRNA translational defect and stalling, which is a potential anti-cancer or anti-disease mechanism. Data that were recapitulated by other groups and other studies. So, but as many of the studies that we have seen in the epitranscriptomics field, most of the data are based on knockout or knockdown. In particular here for METL3, most of the phenotypes were connected to knockout or knockdown. and therefore the true impact of blocking the enzymatic activity in isolation for metal-3 was unknown, and therefore the therapeutic potential of metal-3 remained elusive, at least the therapeutic potential of targeting the catalytic activity of that enzyme, right? So in a lovely collaboration with Storm Therapeutics, a clinical stage company here in Cambridge, we worked hand to hand, and we developed and characterized STM-2457, which, as you can see, I don't know if you can see my cursor here, it's docking really nicely in the sum binding domain of metal-3 in this X-ray crystallography plot. And at the bottom, I'm just showing you that this is a small molecule that shows excellent pharmacological properties with on-target effect on metal-3 in terms of inhibition, as well as on-target biological effects like reduction, strong reduction and selective reduction on M6A, measured by RNA mass spectrometry, as well as reduction of the protein of some of metal-3's M6A substrates like BRD4, MYC, and SP1, known oncogenic proteins. So the first thing that we did at that time was to understand, the true bona fide metal-3 dependent substrates, M6A substrates, and therefore using MON13 AML cell line, we performed methyl RIP and sequencing in vivo, and the STM-2457 treated MON13, and we identified 4,666 peaks that are associated to reduction of M6A upon treatment with 2457. We also observed that the reduction was coming from showing a canonical pattern of distribution of M6A. We didn't see any other pattern or any 5' UTR bias or any other bias. So we were actually inhibiting across the M6A spectrum. But also at the same time, when we looked at structural information and we performed motif analysis, we found that the top 3 significant hits in our motif analysis were 3 different versions of the drug motif, which is the motif, the M6A motif associated with metal-3 catalytic activity, indicating and further highlighting the selectivity of this small molecule. And on the right here, you can see some snapshots of key leukemogenic substrates of M6A, oncogenic substrates like SP1, MYC, and HOX810. You can see in star here, the M6A peak is dropping upon treatment significantly, just the flavor of the different substrates we were inhibiting and the potential relevance to the anti-cancer phenotype we were observing. So as I told you, with genetic disruption of metal-3, we saw that there is an observed and significant mRNA translational defect. So we were not, when the transcript were losing the M6A modification, there was no impact on their transcript on their overall levels, but there was impact on their subsequent mRNA translation. So here we just validate, I'm just showing you for presentation purposes, SP1, one of the M6A substrates of metal-3. You can see that inhibition with 2 different doses of the metal-3 inhibitor didn't show any impact on the overall transcriptional levels. But then when we looked in a polysome profiling experiment, we saw that the transcript levels of SP1 in high molecular weight polysomes were significantly reduced and significantly increased in low molecular weight polysomes, suggesting that we have mRNA translational defect particular to the M6A substrates. And you can see the outcome by a Western blot. You can see in a dose-dependent rates, the protein of SP1 goes down, but the transcripts not, further validating the initial observations using genetic inhibition of metal-3. So being very confident about the efficacy and the molecular activity of this small molecule, we went on and we performed a number of animal studies. Here, I'm just illustrating you 2 PDXs that we used with 50 mg per kg intraperitoneally for 2 weeks. You can see that in both PDX model, patient-derived xenograft models, we have a very significant impairment of AML expansion in vivo, as well as a significant prolongation of the lifespan of the 2457 cohorts in both PDXs, indicating that this is not just a very potent small molecule, is also truly bioavailable and on target. At the same time, and to add an extra clinical relevance in those observations, we looked whether we are impacting not just the bulk cancer in vivo, but also whether we are affecting key cancer stem cell population, leukemia stem cell populations that are associated with aggressive phenotype in the clinic, as well as chemoresistance. And as you can see on the left, by flow cytometry, we can see that in the engrafted leukemic bone marrow, we have significant reduction of the CD93 subpopulation, but also we confirmed that that's true because when we performed secondary transplantation from animals that were treated before with 2457, you can see that here in the intermittent line in red, the bone marrow that was transplanted in the secondary during the secondary transplantations of the treated cohort led to a reduction, to an increase of the survival, indicating that we have definitely reduced the burden of leukemia stem cells, and therefore we saw a latency of engraftment in vivo. So the next thing we question also is that, okay, we see anti-cancer activity and the leukemic activity. What happens on the normal hematopoietic stem cell and progenitor level? Do we affect normal hemopoiesis? So by performing exactly the same therapeutic schema for 2 weeks, 50 milligram per kilogram intraperitoneally in wild-type black 6 mice, we harvested bone marrow and we performed flow cytometry, targeted flow cytometry, as well as single-cell RNA-seq. This is a project that we recently published in collaboration with the Godkin's lab. And the first observations that we saw by targeted RNA mass spectrometry and flow cytometry was not only that we had on-target effect on M6A and selective one, because other relevant modifications appear to not be impacted, like M62A and others like M7G we measured. We have them in the study in the paper. We also saw that we saw the opposite effect from what we saw in the one that we are causing targeting the leukemia stem cells. Here, the normal hematopoietic stem cells are not only were not impacted negatively, we were actually increasing some of their levels positively, which was really interesting. And then to understand whether, okay, we are not affecting the primitive compartments of hemopoiesis, maybe we affect some of the downstream progenitors. So when we performed single-cell RNA-seq, this is how the single-cell 10x RNA-seq single-cell RNA-seq is looking when you subcharacterize the different compartments of the normal hemopoiesis. But on the right, you can see that in blue, this is the branch of the cells that we were reducing upon treatment. And this is the branch that later on we realized that belonged to the erythropoietic compartment. And in red, you can see this is the compartment that we were increasing, which was connected to the hematopoietic stem cell compartment. So we increase hematopoietic stem cell compartments, but we decrease downstream compartments and progenitors that are associated to erythropoiesis. And some sulfate probability calculations that we performed so that confirm that you can see that from the MEP progenitor, the megakaryocyte erythropoietic progenitor, you can see that we have a reduction of the erythrocyte population and an increase in the megakaryocytic population. So in principle, reducing M6A is decreasing erythrocytes, but is increasing platelets if it's happening in a steady state level. And we also managed to see that this is a sulfate event that is occurring even at the hematopoietic stem cell level, so really early in the hematopoietic branch. And finally, we were able to confirm that this is happening just by looking at the blood counts of those animals, vehicle versus treated with an inhibitor, where you can see that we had no effect in overall white blood counts, but we had a slight increase, significant though, on platelet counts, but more importantly, a decrease in our red blood cells as well as hemoglobin. So we were causing a slight anemic effect. So Now, is that a permanent effect that we're causing, or is it something that is reversible? So when we looked at these animals after stopping the treatment a month later, you can see here that not only we didn't cause any impact again on normal hematopoietic stem and progenitor cell pools, but just by looking at the normal blood counts, we actually have a correction towards normality. We didn't see any anemia anymore. So we saw that M6A inhibition is causing a transient anemic effect, which is actually reversible upon withdrawal of the treatment. So to summarize, this was the first study that was showing, demonstrating in vivo activity of an inhibitor of an RNA methyltransferase. It appears to be the proof of concept that RNA-modifying enzymes represent a new druggable class for anti-cancer therapeutics and other diseases as well. So that targeting leukemia stem cells, which was new, is a key mechanism by which metal-3 inhibitor affects leukemias and potentially other cancers. And also the last thing is that a successful small molecule called STC15 entered clinical trials, phase 1 clinical trials a year ago, more than a year ago. I'm gonna say a few things about this as well. So talking about this, I just want to show you a little bit about this clinical stage small molecule to understand a little bit how big is the leap in terms of efficacy and the upgrade in terms of medicinal chemistry. So this is one of the PDXs that we use, and I'll show you for 2457. You can see that with STC15 in vivo, 3 different doses cause a fantastic, a very significant overall survival, an increase in the overall survival compared to vivo. And if you want to actually see how it's comparing to the small molecule with recently published, this is how it looks like. So this is like a day and night in terms of the ability of the small molecule to have an efficacious anti-cancer effect. And also at the same time, if you look within the leukemic bone marrow and the spleen, you can see that we actually affect not just the bulk AML material, but again, we're affecting significantly different subpopulations, different leukemia stem cell populations that matter, the ones that are associated with aggressive phenotype and chemo resistance, like CD93 that I'm showing here, and in a dose-dependent range, especially in spleen. And more importantly, and if you're interested, in 10 days from now, during ASCO, we are going to report some of the results from the phase 1 clinical trials, which is occurring for patients with advanced malignancies of clear unmet medical need. So stay tuned. So the lab is not only focusing on mRNA modifications, it's focusing on tRNA modifications as well. And here I'm gonna just show you a recent collaboration with the Gregory Lab from Harvard University. We focused on metal-1. This is another RNA methyltransferase that is catalyzing M7G and is together with the WDR4, which is the cofactor. Here's the lovely structure that published recently by the Gregory Lab in Nature, showing the full structure together with the tRNA. And that was actually very helpful because then, during our study, which was actually happened earlier than that, we saw that we identified metal-1 as heavily amplified or upregulated or heavily mutated in a number of aggressive cancers. Most of them have unmet medical needs, and that suggested that metal-1 might be a novel epitranscriptomic oncogene. So the first thing we did is using a mouse embryonic fibroblast, we overexpressed catalytic active and inactive version of metal-1 together with WDR4 or an empty vector. We show that the catalytic active version led to a significant increase in colony formation, potentially indicating, boosting of tumorigenesis or potential oncogenesis. The catalytic active version also increased the proliferation of MEFs compared to the other 2 controls, and also increased or boosted the tumor formation in vivo, again compared to catalytic inactive or an empty vector subgroup. So, and then we wonder, how is that driven? How this oncogenic phenotype is driven from the catalytic point of view. So firstly, we identified that overexpression of metal-1 led to a increase in the expression and the stability of a subset of tRNAs, which was then validated, as you can see here. We targeted some of those tRNAs, including arginine TCT. You can see upon overexpression of metal-1, you have a dramatic increase of this tRNA, which we also confirm a significant increase of M7G on the tRNA fraction. But at the same time, we also confirmed that the tRNAs, the subset of tRNAs that were appearing to be stabilized in a gene expression in the RNA-seq, were also appearing to be heavily modified by metal-1, significantly modified when we looked at an M7G RNA immunoprecipitation assay and cleavage performed by the Gregory lab. So finally, to close the circle, we also observed that those tRNAs, obviously, right, are associated with specific codons during mRNA translation, and those codons, when we perform riboseq, plus minus overexpression of metal-1, we saw that upon overexpression, the codons that were associated to those tRNAs were heavily biased towards, they were heavily used, actually, indicating that the pathway towards M7G, tRNA stability, and then particular condo bias on particular mRNAs might be the mechanism of oncogenesis observed. So then we took this tRNA subset and we looked at patient data, genomic data, transcriptomic data, and surprisingly, what we found that many of those tRNAs that are stabilized by metal-1 and modified by metal-1 are heavily upregulated in a number of cancers. And not only that, they were positively correlated with metal-1 levels. As you can see here, we're just following up one of the tRNAs, the one that showed the most striking upregulation and correlation with metal-1, arginine TCT. You can see that their patterns are highly correlated across different cancer models, and that was actually a very strong signal. But to solidify that, we performed SILAC proteomics upon overexpression of either the arginine TCT or the metal-1 catalytic active version, overexpression of metal-1 catalytic active version, and we found a fantastic correlation in terms of targets, in terms of targets that are pivoted in terms of their translation, and those were all targets that were harboring the relevant codons. And you can see here targeted validation of that using overexpression of metal-1 or arginine TCT in mouse embryonic fibroblasts or in leukemia line, confirmed that arginine TCT overexpression actually phenocopied the overexpression effect that we saw with catalytic active version of metal-1. So we actually found what we actually show that metal-1 through stabilization of arginine TCT and other tRNAs drives the aggressive cancer phenotypes or oncogenic transformation, as was validated here in vivo, compared to an empty vector in MOM13 AML line, and which also led, so the higher the aggressive effect and the phenotype, the less the survival rate, as you can see here, which only occurred when we overexpress catalytic active version of metal-1 or overexpression of arginine TCT, but not when we overexpressed an empty vector or a catalytic inactive version. So overall, what we actually show is that metal-1 mediated M7G modification on tRNAs drives oncogenic transformation across different cancers, of course. That arginine TCT, one of the tRNAs that we focus for downstream studies, is a key metal-1 oncogenic substrate, and that upregulation of metal-1 and its tRNA substrates increases growth promoting proteins, known proteins that have known roles in a number of cancer phenotypes. And of course, what we actually show is that metal-1 is a new druggable target for a number of malignancies. And since I mentioned that, very recently, we reported during the American Society of Hematology meeting and the AACR, the first tRNA methyltransferase inhibitor, and again in collaboration with Storm Therapeutics. We call it STM-9005. Again, this is a very potent and excellent small molecule, bioavailable as well, that has a really nice biochemical activity on metal-1. It has a specific on-target effect on M7G, measured both at 48 hours and 72 hours by RNA mass spectrometry, as you can see here, with 2 independent doses. And then using primary urine AML, as you can see that it has also a potent anti-leukemic effect ex vivo. At the same time, I'm showing you here that this is a selective small molecule, because in a RNA methyltransferase, say at 10 micromolar, we only inhibited the activity of metal-1 and none of the other circa 50 methyltransferases that we examined. And at the same time, by looking at a large panel of RNA modifications in RNA mass spectrometry, we confirmed that we only block M7G upon inhibition or catalytic inhibition of metal-1 using 9005 and not any other irrelevant RNA modifications on tRNA like M1A or M5C. So how are we impacting the cancer cells equally as we saw with the genetic disruption of metal-1? The answer is yes. But actually what was very interesting is that we saw that isolated catalytic inhibition of metal-1 led to an increase in myeloid differentiation of leukemia models. Here, 2 human AML, I'm illustrating 2 human AML models, THP1 and OCML2, but also a primary murine AML model where you can see that there was an increase in myeloid differentiation and also in a dose-dependent range, indicating that the catalytic inhibition, as we initially reported, is part of a leukemogenic mechanism or carcinogenic mechanism, either a mechanism of cancer initiation or maintenance. So last few slides, here I can show you that when we performed preliminary animal work, we're using an aggressive primary murine model driven by an MLLAF9 and a FLT3 ITD. This is quite an aggressive AML model. You can see that we, just by treating twice daily with 30 mg per kg in 9,000 with the 9005, we achieved a significant prolongation of the lifespan of the treated cohort here in green, and you can see also that we have a significant reduction of the leukemia cells in the periphery, in the peripheral blood cell, in the peripheral blood, just by treating with 9005 for a week, which was an indication that with just a bit more optimization, we could actually achieve even more striking effect. But the question is, are we just seeing an effect in hematological cancers? The answer is no. We also looked at a melanoma model, a very aggressive melanoma model, which is a model that is resistant to our metal-3 inhibitor that I showed you before. But here you can see that one image is 1,000 words, like 9005 significantly reduced the tumor growth in vivo compared to the vehicle treatment. You can see also the trajectory of the tumor growth per group and per mouse. But most importantly, we were able to validate on-target effect of this small molecule in vivo. You can see that M7G measurement is significantly reduced in these tumors, both when we looked actually within the tumor, but also when we looked at other tissues like kidney. So we have a very significant on-target effect and appears to be quite selective. So to summarize here, I'm showing you the development again of a first-in-class tRNA methyltransferase inhibitor. We call it 9005. We're going to report it later this year. This is a efficacious and a selective in vitro, ex vivo, as well as in vivo in a number of aggressive malignancies. We have many more models that we have been testing lately, and we are currently working also on the molecular mechanism that we see, and we are trying to connect the dots to see which is the actual molecular mechanism of this anti-cancer effect. But most importantly, the takeout message here is that I think in general, finding novel therapeutic ways to target modifications of tRNA could be very beneficial, because tRNA adaptation is one of the most important things in cell fate decisions and disease. So I think I don't have time to cover the rest of the talk, so I think I will just wrap it up here. And I would like to thank these fantastic people in my lab, in particular, our postdoc Eli Yankova, who has driven most of this work, both on the metal-3 inhibitors, as well as the novel metal-1 inhibitor. Our collaborators, the Gregory lab, the Godkins lab, the Vasiliou lab, Tony Kouzoridis and his lab, the Irmela Jeremias, Alan Bradley, Nerea Erigoyen and her lab, our institutes that are hosting this work, as well as our collaborators, most importantly, our collaborators in Stone Therapeutics, as well as our funders that are supporting generously this effort. Thank you very much.
Thank you very much, Kostas. Amazing talk. I will briefly read a couple of questions that we have in the chat.
Yeah, go for it.
The first one is there any potential therapeutic application associated with regulating the balance of M6A in the brain in the case of non-pathologies?
In the case of non-pathologies, there is no at the moment there is no direct evidence, therapeutic evidence that an RNA methyltransferase inhibitor, at least the ones that we have developed, could cause an anti-disease effect in vivo, especially in the brain. However, the reason why I'm saying that is not that there are several publications showing that genetic disruption, RNA methyltransferases and RNA enzymes may impact positively, negatively, disease models associated with brain tumors, for example, or other brain diseases. However, because many of those small molecules that we have developed or they exist from elsewhere are not brain penetrant, we cannot directly test those questions and answer some of those questions. But I think that there is a lot of effort lately from our end and from others to create brain penetrant small molecule versions of what we have shown already. And I think this is where we will directly test not only the anti-cancer effect in brain, but also potentially test the general tissue biology of brain. So I think there are good stuff happening, but yeah, they are still under evaluation.
I guess also the blood-brain barrier doesn't really help.
Yeah, that's what I mean, that we don't have good brain penetrant small molecules directly. So we are relying on animal models or genetic inhibition. Sometimes is good, but sometimes is pleiotropic.
Okay, thank you. Have you looked into targeting specific ribosomal RNA modifications? Do you think it might be an interesting therapeutic target?
So they are definitely an interesting therapeutic target, especially, some leukemias that we're working on are driven by ribosomal modifications. And there are also other ribosomal modifications that are connected to be ribosomal modifications, but under different contexts, they may be catalytic active on other RNA fractions. But yeah, I think that the fact that they are functioning on the ribosomal RNA doesn't mean that they're not very good therapeutic targets. It means that we may have to fine-tune the level of inhibition. And some people think that, if you go with a ribosomal RNA inhibitor, an inhibitor that blocks a ribosomal RNA modification, it might be unessential or super toxic. From what we have seen from the small molecules we are characterizing, it might be the opposite, that you may need to go harder and longer to actually start seeing an effect. So
is it because of the abundance of the
abundance and the turnaround of the RNAs? So
okay, thank you. One last one, and maybe you will answer the other ones later. Did you encounter any specific mutation of background in leukemia that were refractory to either metal-3 or 1 inhibitors, or the other way, mutational backgrounds that responded better than others?
So this is a question, there is a debatable, question. I would say, I'll tell you why. This class of small molecules of, modalities are affecting, as the modifications, right? They are affecting a number of pathways, because there is a number of transcripts that are going to be affected. We're affecting a big number of pathways. And therefore, I think it's unrealistic to say that that's going to be connected to a particular subtype of a cancer of AML. I think that what is more realistic, and that's something that is expanding more in the field of cancer, and especially in clinical oncology at the moment, is we tend to believe that therapy should be associated to group cancers based on that, the pathways they are using rather than the mutations. Because as we move from, pan-lethal treatments like chemo to more specialized treatments, we might have to group them based on, for example, MYC-driven cancers, HOX-driven cancers, RAS-driven cancers, these kind of things. The most important thing, and I would say I will leave it there, is that the first thing we are checking is whether mutations associated with cancer relapse or secondary cancer effects or secondary AML, like TP53 mutations, BAX mutations, other mutations that are associated to, aggressive and refractory cases, whether those are actually reducing the anti-cancer effect of our small molecules. And thankfully for those, that's not the case, at least for the ones we have presented today. So those are actually not enough to cause any resistance, which actually creates a window for us to suggest those potential therapies, either as monotherapies or in combination with, mainstream current treatments.
Yeah. Thank you so much. I think we are running out of time, so I think we should wrap it up. So thanks a lot to both Anna and Kostas for the great talks. And thank you all very much for attending. The next RNA collaborative seminar will be the 5th of June. And so check it out on the RNA Society website. And with this, I thank you all again and hope to see you soon. Bye-bye.
Thank you. Bye. Bye.